This is going to be all about the liver. The liver is a vital organ for our survival and carries out many functions, which are going to be explained a bit later on. But let's start with the liver structure. For a visual reference, turn to page 20 in your CGP A2 biology guide. The liver is made up of liver lobules, which are cylindrical in shape and are made up of hepatocytes. These are arranged in rows, radiating away from the central vein. There are many branches connected to the central vein, such as the hepatic vein, hepatic portal vein, and bile duct. The hepatic portal vein and artery are connected to the central vein by capillaries called sinusoids. So how does the liver structure actually relate to its function? Well, blood enters into the liver through the sinusoids and runs past the hepatocytes, which remove harmful substances and oxygen from the blood. The harmful substances are then broken down by the hepatocytes into less harmful substances, which are then safe to re-enter into the blood. Cells called the Kupffer cells are attached to the walls of the sinusoids, which remove bacteria and break down all red blood cells. These two processes, in a way, act as a cleaning system for our blood. The central veins from our lobules connect up to form a hepatic vein. So let's move on to excretion. Excretion is the removal of waste products from the body. The liver plays a key role in the excretion of excess harmful substances, such as an excess of amino acids, alcohol, paracetamol, and of course insulin. Excess consumption of amino acids and proteins could be extremely harmful to our bodies. This is because nitrogenous substances can't usually be stored in our bodies, so we're left with one of two options. They either have to be used in our bodies to make more proteins or such roles, or to be broken down and excreted. We're interested in how they're broken down. So firstly, the nitrogen-containing amino acids are removed from any other excess amino acids, forming ammonia and organic acids. This process is called deamination. The organic acids go on to be respired to give ATP, or convert into carbohydrates. Ammonia, on the other hand, is far too toxic to be excreted directly, therefore it's combined to CO2 during the ornithine cycle to create urea. This is then released back into the blood and filtered out by the kidneys at a later stage. The liver also removes other harmful substances from the blood, such as alcohol, drugs, and excess hormones. So, if you're like all of us, you may have overdone it at the weekend. Your liver is going to have to work overtime to get rid of all that extra alcohol toxins which could damage our cells. So to do this, it firstly breaks down it into ethanol, which is then broken down even further into less harmful substances called acetic acid. However, if your liver is exposed to too much alcohol over a long period of time, it can lead to cirrhosis of the liver, which ultimately leads to your liver cells actually dying and causing scar tissue, which blocks blood flow. So to combat your hangover, you're going to take some paracetamol. But again, the liver has to deal with this, and too much can lead to liver and kidney failure, which is a major problem. The final substances that liver breaks down is insulin. We have to do this as excess insulin can cause a problem when trying to control blood sugar levels. So, that's about it. To recap, the main roles of the liver is involved in excretion. It's made of liver lobules, and it breaks down harmful substances such as amino acids, alcohol, paracetamol, and insulin.